I'm sure that you all heard a lot about what is machine learning, and the general idea here is to design algorithms that enable to learn from the prior experience. And now, what does it mean exactly?
So, can I start sharing? Can you enable me to share screen? Hello?
Yes, sir. Yes, I will. Yes. Now you can share, sir. It's enabled.
Yeah, thank you.
Yeah, thank you, sir.
So welcome everybody. Good morning. So today we will be discussing about MIT. MIT, one of the top institutes in the world. They have got a consortium. Okay. So this consortium is actually called as the Machine Learning for Pharmaceutical Discovery and Synthesis Consortium. Okay. So otherwise called as MLPDS. So Machine Learning for Pharmaceutical Discovery and Synthesis Consortium. So here a group of scientists from MIT, they are collaborating to write machine learning codes and these kind of web pages, web servers, okay, develop web servers for actually developing tools, okay, by helping the people to actually use machine learning for machine learning for actually like accelerating drug discovery by using it at various stages. Okay, for example, from molecular property prediction to even reaction outcomes. For example, if you take two reagents and in the process of synthesis, you try to come up with a product. So alternate synthetic routes can be tried. And then those cases, even the products, the byproducts, the side reactions, all those things can be predicted now. Okay, so now even organic reaction outcomes also can be now predicted. Similarly, they have developed about stereochemistry, stereochemical handling. Okay, so they have come up with a neural network which can actually guide you in getting the correct stereoisomer. Okay, because in pharmaceutical drug discovery, pharmaceutical drug synthesis, the drug actually can be an enantiomer. The drug can be actually active in one of those, R or S forms. Okay. So the thing is like to guide the synthesis in the correct to get the correct SMR, again, machine learning has been used. So here, this machine learning for pharmaceutical discovery and synthesis consortium, they help in synthetic planning, they help in property prediction, they help in reaction outcomes and molecular graph generation, stereochemical handling, on all those things. Now we will see a small video about one of those. For example, these are the people working in that machine learning for pharmaceutical discovery and synthesis consortium, out of which Dr. Regina Barzilay, okay, she's one of the top scientists there. So she's a professor there at MIT, and she has been active in developing machine learning codes, okay, and developing other tools, web-based tools, to enable people to use ML-based, predictions about, drug synthesis outcomes and all those things. Now we'll see a small video about her lecture, okay. So a small talk by Dr. Regina Barzilay from MIT.
Okay, to learn. To learn means not to memorize. So if I am trying to give you a recommendation of the product and I have all the past experience, the idea is not to find somebody who is exactly like me, but to find somebody who is similar. And the whole question is, how do we come up with a good mappings that enable us to generalize in a smart way? So let's make it very concrete and start with a question about, again, learning to recommend. So here what you can see is a very simple task. Let's say we're trying to predict which movies a particular user would like. And we have the set of users, the set of movies. And for each movie during training, and you see it on one side, the training, we would know exactly the answer. We would know these movies the user liked, these movies the user didn't like. Okay, I didn't select those movies. It's Tommy Yakala's selection. And at the end, you have a new movie, and you need to decide whether the user would like it or not. Now, of course, machine cannot look at such complex object as a movie and just decide whether the user likes it. Typically, the first step that we do in machine learning, we design feature vectors. Or for many of you, you call them variables in statistical experiment. You're deciding what kind of factors are going to be the one driving the predictions. So for instance, in the case of movies, it could be the genre, the producer, the actors, if the movie has a happy ending, whatever features you're thinking about. And every place in the vector would correspond to a specific question. For instance, the first place would record 0 or 1, depending on whether the movie is an action movie or not. And it's up to you when you are using.
So here she is actually trying to give an analogy, okay, an analogy for molecular vectorization. Okay, so here the feature vectorization is she's talking about molecular feature vectorization. So different features about a molecule. Okay, so so people actually do 2D structure fingerprinting, and from that they get the molecular vectorization. Okay, so she's comparing that one with the movie. Okay, so how people choose movies based on actors, based on the film, based on language, like that. So it's given an analogy. Okay,
using machine learning to select which features can be used. Of course, you should be able to compute them, but you have a freedom how to decide about the features. So at the end, what we are doing here, again, this feature representation step, we are taking an object and translating them into a vector, where every coordinate in the vector record a specific property of that object. So now, if we look again at this movie, what actually machine sees, it sees a vector and it sees a corresponding label, +1 or -1. So in this case, you can really see that we can, in the same representation, do books or do molecules or do something else. Machine would see vectors and the corresponding labels. And our goal is to use training set where we know the answer to learn the correspondence between the vectors and the corresponding labels, to use it to be able to classify unseen examples during test. And in this case, you know, as I told you, we have a freedom to use any number of features that you want. But just in order to give you a different feeling what machine learning does, let's consider an example where we would represent every movie with just 2 features, okay? And the reason we are making this simplification because I want to draw it for you in 2D. I cannot draw it to you in 10D, so we'll focus on 2D. So actually what you see here is now every movie is a point in a space. The first coordinate would record the value of the first, you know, of the first feature and the second coordinate of the second feature. And each one of them comes now with pluses and minuses, correct? And what the machine learning model needs to do is actually to divide the space of pluses and minuses. So geometrically, you can say that if I take my whole space and divide it into 2 parts with pluses and with the minuses, I solved my machine learning problem. I mean, we don't know how well you solved it, but you did solve it. Why? Because now if you take any point in this space, you would know what kind of sign you should put to it. So in this particular case, for instance, our movie in the test case would be in the same place where all the positive examples are located. So we will have all the positive on one side and all the negatives on another side. And I would show you some other representation of the same idea. Again, we're looking at it from all the directions so we understand it better. And we will need this representation when we talk about deep neural networks. So what I drew you here, it's exactly the same feature vector, correct? And when it's black, it means that we have, let's say, 1. In all the places, it's 0. You can see that here the first coordinate is PG13. In this case, it's on. And what machine learning will do, it would learn what weights do we need to have for each of these feature values in order to combine them to give you a score. And according to the score value, you would classify it as +1 or -1, okay? So the next point that some of you may be wondering, you're thinking, okay, the example that you showed to us is very simple. We can actually perfectly separate all the pluses and all the minuses, but what happens if pluses and minuses are really intermingled and there is no linear classifier, no line that can separate them? This is very correct, and that's why we're going to be talking about complex, non-linear classifiers like deep neural networks, which would be able to deliver you this capacity. But at this point, this is just a detail how these boundaries would look like. And I'm sure many of you heard about variety of different machine learning models, deep learning models. So those of you who would know maximum entropy, many other classifiers, you were wondering, how can we think about all these different models in a general way? What is the connection between all of them? So any machine learning models try to optimize 2 objectives, which are kind of standing one against another. The first objective is to fit perfectly your training example. Not perfectly, but to fit well your training examples. Training examples is the only truth that you know. Because if you have to learn mapping from something, it will definitely be training examples. So then you can say how well I am doing. And that is, you know, maybe the way we would score like a homework or something, we'll say how many correct and incorrect answers do you get. This is an obvious way to score it. For machines which are doing optimization, it may be not the optimal way to count it. So there are other ways you can count it, you can look how far they're from the boundary and use some other measures. But the first criteria is that you optimize how well I feed the training examples, how close I am, however we define the word close to the training example. So this is an obvious thing. The less obvious thing is actually to keep the model simple. Why do we want to keep the model simple? Because like look at the picture, you see 2 different separators. One is a straight line, another one is something very curvy. And you can say, maybe I should be using something very curvy. But what we know is that typically when you feed the model, you have limited number of training examples. And more complex is your separator, more parameters you need to train. And you have much higher chance of overfitting if your separator is extremely complex. And also your data may have some noise. So if you're perfectly optimizing every single example, maybe it's not a good thing for generalization. So there is typically, it's called regularization. This is a measure how simple is your model. And there are many ways exactly to capture it. Now, if you are looking at any objective for any machine learning model, what you would see that this objective combines the loss, our agreement with the training examples, and this regularization, which captures how complex is our classifier. And once you decide about the objective, you set up an objective, the second important component of the algorithm is optimization procedure. Because even you decided that now I want to have straight lines and that's how I measure my loss, I need to go over the set of all possible straight line and find the good one. So, and there are many ways to do optimization, for instance, stochastic gradient descents and many others. And typically whenever you are selecting the objective, you need to find an optimization method which works well with it. Now, you don't need obviously to do any of that because today there are packages which do it for you. But the reason I'm talking about this is that you can understand the space of possibilities, and you can see on the slide, you know, variety of classifiers that are commonly used today in practice. And let me now give you concrete examples of how we can use this very simple classifier, even a line kind of a linear classifier, to solve real tasks. Because as users of machine learning, your goal is not to design objectives or to find the best optimization procedure. It's only given to you in the package, is to think, how do I take my problem and translate it into machine learning terms? And I will show you two examples, one from natural language processing, another one from chemistry, so we can understand how it could be done. So the first one is to take natural language document. This is extract from pathology report, and to predict whether it describes a malignant case where there is a cancer or not. Of course, machine cannot read natural language the way we do and fully understand it. So we need now to take this model and translate it into a way which will be understandable to our machine. So what we are going to be doing is creating representation which is called bag of words. So what we will do, we will take a document which has sentences, which have all beautiful structures, and destroy it. We are going to now take this document and represent it as a vector, where every position in a vector would record presence or absence of a particular word. For instance, the first position would always record whether the word patient is present or absent. And the second position here would look whether the word estrogen is present or absent. So you can see that now I took my document and recorded it as a vector. And let's say I can count how many times it's present or absent. And of course, I lost a lot when I did this representation. Why did I lose? For instance, now any random permutation of my document would result in exactly the same vector. But let's just stick with this representation and see how far we can go. So now again, we took all our documents, created corresponding vectors which record presence or absence of the words. And here you may be thinking to yourself, which words should I put? How big is my vector? So take all the words in English, make it huge, you know, if you want, or take all your pathology reports, see what vocabulary, what words are used there, and then create a vector. However you go about it, you may get a very long vector. And again, you may be thinking, and what happens if I want to record the word like note? Note on its own doesn't say much. It depends what note modifying it. So let's just resolve this problem by adding bigraphs. So not only we're going to add individual words, we're also going to get pairs of two adjacent words, or let's say triples. It will dramatically increase the size of your vectors, correct? But you can still, for instance, cut out triples or bigrams which appear less than 5 times or something. Okay, so at any rate, we took the document, translated it into this representation bag of words, and now we're exactly to the same position. We have our vectors, we have labels, and we can use our classifier to actually separate them. And I can, again, what the machine would learn, the same view as before. We have this very long, sparse feature vector representation, machine learn the weights and makes a prediction. And what I want to show you is which words machine actually used for specific prediction. So what you see on the left is a classifier trained to predict invasive ductal carcinoma, IDC. And you can see that the wards which are high predictors of this disease are invasive ductal carcinoma, not surprising, infiltrating ductal, excision invasive ductal, and so on. The wards which are shows that there is no IDC are the wards like no evidence of residual invasion node and so on. And you can do, observe the same for the other disease LCIS. So this is something that machine learn without any touch of a human. Human just provided machine with training examples, and then machine learn what are the features, what are the words that are predictive of certain class. Okay? And then we are bringing ourselves to another very important question in machine learning, especially in applied machine learning that you're going to be doing, is how much training data you actually need to have, correct? And this is a big question because, it typically costs money to annotate the data. And it's not even clear that if you don't have enough data, you may be unable to do good work. So the way people typically do it in practice is using what is called learning curves. So what you will do, you will take the data which you already have and record performance, train your model on a subsets of the data. So in this example, we would, let's say, train it on 10 reports, on 100 reports, on 500 reports, and so on and so forth. And you can see how the performance changes when we are increasing the amount of data. It's surprising that, of course, it increases. The point is how fast it continues to increase. You can see that when we are moving like from 100 to 200 example, the slope is pretty steep. The model increases very fast. It's really hungry here, needs more examples. However, when we go to 500 examples, they increase to 1,000 and not that dramatic. So if it really costs you, maybe you don't need to annotate another 1,000 example. And the reason, if the model underperforms, can be maybe wrong features and other things. It's not the amount of data that is going to fix it, okay? Let's now move to the domain which is very familiar to you and less to me, it's chemistry. Let's look at the tasks that many of you deal in the daily basis, predicting chemical properties. So in this case, you're given the molecule and let's say you want to predict toxicity. So there are several modeling questions that you need to be asking yourself. And I know that there are ways for certain chemical properties like log P, which you can compute analytically. And if the analytical model works well, there is no point to change it. We're talking about the properties where we cannot do precise deterministic computation, where we really want to combine many sources of evidence and help us do prediction. So the first question for you is what training testing data I'm going to be using. So you need to find a question for which actually you do have data. The second question that you want to ask yourself, what are the appropriate labels? So as I already discovered by talking to many of you, that just asking toxic or not toxic is not the right question. You may want to ask in which dosage it is toxic, which organ it is toxic. So those are all great questions. You need to decide exactly which one of them you're going to be modeling. But of course, you need to have training data to answer this question. And finally, is what representation we are going to be using. Clearly, in this case, we cannot use bag of atoms approach because it's going to lose all the structural information about the molecules. So the first option of bag of atoms is done. The second option that many of you are using in practice is Morgan fingerprints, correct? Where we take molecule and we translate it into particular substructures, and now we can represent molecule over the space of these substructures. So there is advantage and disadvantage in using the representation. The advantage would be that whenever Morgan fingerprint was constructed, it based on kind of chemical understanding of domain experts, and it captures maybe many important features that that really drives the properties of molecules. The disadvantage of it, that this representation will be the same no matter what properties you are trying to predict. And maybe for some properties that you're trying to predict, certain constructions are more important than others. So what we will see later on in my presentation is actually how we can do custom land representation. We will see a neural alternative to Morgan fingerprints, but for now, let's just stick to Morgan fingerprints, okay? So again, what we're doing here, we're taking molecule, and we are looking, all different substructures that produce from Morgan fingerprint, whether they're present or absent, and then we can compute the score. So as you can see now, we have machinery which can, given the sufficient amount of training data, can ask many property prediction questions. So let me summarize this part of the presentation and talk to you about what are the essential steps in supervised learning. The first point is you need to convince yourself that there is patterns in the data. And since all of you are domain experts, you typically know if the pattern is there or not. And again, if you can analytically compute something perfectly, there is no point to do machine learning. The point is that there is a pattern, but it's not strong enough, or it's too complex to write it deterministically. You want machine to learn it. The second question is to decide which features to use. And this is an important question because if certain information is not provided to the machine, it would never know. Like, let's say you're trying to predict some high-risk population. If you didn't put information about family history, there is no way for machine to recreate it. So the algorithms that we are looking on here, they are good with dealing maybe with redundant features, but if the feature is not there, we will be unable to compensate for lack of it. And finally, we need to collect training data. The more training data we have, the better. We need to be aware whether we have noise in the data. And if the data is incomplete, there is some systematic bias, it will definitely negatively impact the learning because typically garbage in, garbage out. And the final piece, we need to evaluate our model very carefully. And typically the way we do it here is we have a training data, we have a development set and the testing set. What does it mean? The test set we never touch until the end, until we really are done, that there is nothing to be done. It's like really unseen, like a test in my machine learning class, students do not see it ever until they're done with the class. Development set serves as a proxy for the training set, because for the test set. So you can try a variety of things, you look at different models until you convince yourself that you have the best possible model. And then you can, when you are totally done with your development, you can really try it on your test set. And it will give you the idea how well the model will score when you give it totally new molecule it hasn't seen before. And of course, another important point that I want to make sure that we understand, and this is a real question for people who work, with the new chemistry. The question is how similar should be the molecule that you are testing on to the molecule that you actually used in training. And this is a real question because these models work well when the training set distribution matches the testing set, the case of application. And later on, in one of the lectures, we will talk about more complex models, which are called transfer learning models, which enables you to scale. But if now you have a training and testing data, which is kind of similar, you can use these techniques pretty reliably. Okay, so now we are going to talk about the next exciting moment. It's about deep neural networks. And deep neural networks now really became, the strongest classifiers that are available. And if you have, sufficient amount of training data, go ahead and use it. But let's understand what do they do. So here, what you see on the picture, with the circles, it's actually a drawing of neural net. The first layer is your input layer, the layers that we've seen where our vector comes in. The last layer is the output layer. And what you've seen in between are layers of processing units. So the idea in deep neural networks is that we take many very simple processing units, string them together in some smart way, and as a result of it, we will provide it with the capacity to identify extremely complex patterns that our simple processing units couldn't do. So it's really the analogies that I give, it's like ants. Every ant is very simple, cannot do much, but if you take the whole colony and coordinate them, they can do great things. And originally it was developed in the context of neurons, of brain neurons, because neuron is a simple processing system, which takes an input or inputs and generates you an output, and they're all combined together to give you the prediction. So here is the idea, but so far, that's where it ends. So there is very little connection between deep neural networks that we're using today and what's going on in the brain. And the reason they're called deep, because typically you have multiple layers of these transformations. So let me just tell you, and this is the question that people constantly ask. You know, I've heard about neural network like 30 years ago, 20 years ago, depending on your age, and it was like dead end. How come they become so popular? And it's very true. You know, when I was PhD student, it was considered like to be totally dead topic that you shouldn't be touching. So how did they become so prominent? And actually, the point when it became prominent is in 2012. When people who were producing neural network competed in ImageNet competition. So this competition contains 1 million images, and you can see some examples. You can see the images are really not trivial. For instance, look at this picture on the bottom. I would say it's a dog, but it's apparently cherries. And there are 1,000 different categories on which machine should do the prediction. Here, what you can see the performance over the years. So in 2012, all the models but one were not deep learned. They used kind of more handcrafted features and filters to do predictions. The very red one comes from Jeffrey Hinton from Toronto that used deep neural network to train it to this prediction task. You can see that the error was 15.3%, which is way better than anybody else who didn't use deep learning. But what is more remarkable is to see what happened in 2013, where we can see that this performance was actually at the very bottom of the next year's system, and now our accuracy, our error is 11.7. And again, the same pattern is repeated next year when we see that now the top performance in 2013 becomes the bottom performance in 2014. So we can really see that this complex model with millions of parameters are really driving the performance and solving tasks which previously were considered very hard and unsolved. So the question that people ask, what happened? And there are 4 reasons. Some of them have to do with environment, and some of them have to do with our algorithmic understanding. So first of all, in order to train neural models, you need to have a lot of data. What it actually tells to some of you that, like for instance, when we're talking about clinical trials, we may not have enough data to run neural models. There are other models that you could do, but not directly take neural models out of the box and run it. The second point is computational resources. Now we have GPUs, graphical processing units, which are extremely powerful, and you need to have them. And this technology just became available relatively recently. The third point that people discover, and it has to do with algorithms. So the first generation of people who work on neural networks had this misconception, which is actually a reasonable scientific hypothesis, that if you want to make the model work, let's first make a very small model work. And small, we mean very few layers. And what people found out, it's actually much easier to make a large model work than to make a small model work. And this was a change in the amount of compute and of data enabled to train these large models. And the final reason, which I think has a very high relevance for all of you, is the fact that today, luckily for all of us, is that all the machinery of neural network is actually encapsulated into these big Lego pieces. You don't need to design all the, kind of tedious details of how to train this model and to compose it. You have big blocks which you can assemble together very easily and customize it for your application. And that's what actually drives democratization of this technology to people who are not computer scientists or not machine learning experts. So let me just show you again neural network. Again, as you've seen here, I like to show pictures of the same network. And I should say that this particular drawing was inspired by the drawing that I saw in Jay Bradner's presentation. So it's a nice picture. So here, again, you can see our input layer, the one that we use for linear classifiers. And then what we have, it is connected to the hidden layer, the one in the middle. And how it is connected, every one of these edges has a weight, okay? It has a weight. And then within the hidden unit, we will have some non-linear transformation. That's what enables this model to to generate this very nice nonlinear boundaries is the presence of nonlinear transformation. It can be sigmoid, it can be tanh. There are many different options here. And then again, we now took our original vector, computed the next transformation within the hidden layer. And in this case, we just explicitly connected to the output layer, but we can pipeline and have more hidden layers. Okay, so there are two reasons why people love neural network. One is, of course, as we've seen, they achieve superior performance, and this is great, it's just a better classifier. But there is another reason that I want you to understand, which I think has a lot of interesting potential in chemistry, is actually learning representation. And now in the second part of the talk, I will, of my tutorial, I would actually talk what they mean by learning the representation, okay? So let's get started. Let's go back to what we discussed previously about bag of words approach. So let's think what we've done. Whenever we took the text with all many words that this text contained and represented it as this vector which records whether the word is present or absent. Another view on this representation is to say, now every word in my document is just a vector. And for every word in the vocabulary, for every word type, I'm going to have a position. Let's say the word I would always be on the first position, okay? And the word love will be in the second position. And now every single word in English will have position dedicated to it. What does it mean? That my vectors would be on the size of the English vocabulary, let's say 20,000 size vectors. Very big sparse vector where it's just one hot. There's just one place which would have one. Now, how did I get a document out of this representation? I just summed up together all these one hot encodings and I got all my bag of words. So this is good, but there are two concerns that we may be having here. One is it the best representation for word as like very long sparse vector with one on or off? Is it the best representation? The second one is it the best way to get a text just by summing up the vectors? Can't we do better? And let me start with the first question. This is a question of representation, okay? So we have two questions, representation and the combination. So let's start with the representation. Let's say you're training a classifier which tries to predict whether your document talks about dogs or not, okay? So you take a sentence, I love dogs, it has the word dogs, and if your model have a lot of examples, it would hopefully learn that, you know, if the word dogs is there, then it means that the document is about dogs. All good. Now, assuming that during test, you are going to have a sentence which says the Samoyed is lovely. This is a type of dog, big white dogs. Okay? The model never seen the word Samoyed in its life. It doesn't know that the word Samoyed and the dog are related in any way. What is it going to do? It's going to misclassify this example, correct? Because it doesn't have our semantic understanding. The dog and Samoyed are actually similar. So what we want to do, and this is really remarkable and interesting point, is to learn representation, which reflects the semantics of the domain. Now I'm talking about the semantics of natural language. Very shortly, we're going to talk about semantics of chemistry. So what I want to do is to take this very big, sparse, one-hot encoding of the words and translate them into dense, continuous vectors. And the only criteria that I would care about is to preserve the semantics. What I mean, I want the words dog and Samoyed to have similar vectors. Similar vector means that they're close to each other, while the word apple, for instance, should be farther away from them. It should have much bigger angle from these original ones. So we want to have this vector in such a way that close words are close to each other, semantically close words, and the vectors of the words which are very far away should really be far away. And this sounds like really a tough thing to do because we definitely don't want to make machine to teach it the whole English language, correct? Explicitly at least. So how can we do it? Very simple, as it turns out. So what we will do, we will now forget about specific tasks. We are going to take a billion corpus of English language, which would have sentences, maybe from tweets, maybe from newspapers, whatever, very, very large corpus. And we are going to train it on a task, let's say, of predicting the next word. So let's say if I give you a sentence, I go to Barisma to get my morning, you know, the word coffee or tea should have like higher likelihood that, let's say, potatoes. So what we will do, we will train the model to predict the next word. This task is called language modeling. So you don't need specifically annotated training data because text give you a lot of training data. So this is fine. You can train the model, you know, it will do it with certain accuracy. But what is more important, that in addition to these models that predicts it, you can actually take, let's say, the first layer of this model, the first layer of the network, and say, now this first layer would correspond to my word. I mean, you know that for every word, when you run through it, you're going to get different representation in the first layer, because each one of them will activate the network in a different way. And from the intuitive point of view, the words which are closely linked semantically, like a dog and Samoyed, because they appear in similar contexts, because their prediction should be similar, the corresponding weights in the network should be very similar. That's why whenever these words which are similar on this task activate the network, the layers, values that they produce should be similar to each other. And similarly, the words which are very different from each other, because they give different prediction when we're predicting context, should look in a very different way. So there is a whole cottage industry on creating kind of models, which layers should we do the first one, the last one. Those are details. The most important point is to understand that when we are doing this auxiliary task, we can take one layer of the network and use it as an embedding. It would be continuous and it will be much smaller in terms of coordinates than the original vector we are feeding. And I just want to illustrate you that this is not a crazy idea. So you can see here a 2D projection. Okay, we took these multidimensional vectors and we projected them in 2D because we cannot again see stuff in multidimensional space. And now you can see what are the words, each word represents a vector. And you can see that the vectors, the words which are very close, they do represent a lot of semantic similarity. For instance, we have here kitchen, vanity, sink, bathroom, all this kind of related word. This is our blue cloud. And there is a word like charger, battery, whatever, Bosch, which represent the brown cloud. So this is what machine learned without ever understanding semantics of English language. Never, it never seen any, we never put it. That's what machine learned by looking at a very large corpus, doing this context prediction task, and by taking out the representations. So this brings us to the next important question, which is how we can go about encoding molecules. And again, what we've said previously, we can just take Morgan fingerprint and use it, but maybe we can do something better. So one way, I will show you truly better way, but this is kind of intermediate better way. You can say, okay, I am gonna take my molecule, translate it into Morgan fingerprint, and then run my neural network to predict, let's say, toxicity, to predict some property. And then I am gonna, once I trained my model, I am gonna take, let's say, the first layer and use it as a representation of this molecule. Like for human, which looks at it, it will make no sense. In Morgan fingerprint, you know exactly from which structure each coordinate came, but here you just wanna have a dense representation. And here my student Van Gogh actually visualized for us what the model actually learned. And given my lack of chemistry knowledge, I comment on it, but I was assured by many that this actually, the connection between this molecule really captures meaningful properties. And again, this particular model doesn't understand anything about chemistry. It was just trained, we learned the representation, we showed the projection, and supposedly these molecules, which are close together, really exhibit some similar properties. Now I want to go, so we talked about representation, and I will revisit this question later, but I want to talk about very important question for us, both in language and in chemistry, is how we can actually combine them. Because what I told you, there is a way for us to create representation for words, but how do I build representation for a document? And previously, I gave you a very simple answer, just sum them up, not very satisfactory. Maybe we can do better. So, and the interesting part here is that now everything is in a vector space. Every word is a vector, and the document is a vector, correct? So how do we go from the vector words to the sentence word? And here, I will give you just one example of a very powerful construct, which is called recurrent neural networks. For instance, maybe some of you have heard LSTM, which actually provide us with a very efficient way of representing context. The problems that we observed when we were just summing up these vectors that we've done in the past, the problem with that was that we're actually discarding the order. We're discarding how the words are connected into the sentences, correct? And we are losing a lot of information. So here, the idea is to actually combine these different unit representation together, taking context into account. And if you remember, I talked to you about Lego pieces. So here you see the first Lego piece. So that Lego piece, a small computing device, like a small neural network, what it does, it takes two inputs. One is, let's say, a new word and the context, the previous context. And what it does, it just compute and generates you another context. So let me make you a very concrete example. Let's say we are processing a sentence, I have seen better talks. I should have said I have seen better tutorials. Okay, so in this case, we start originally with a null context. There is no context, it's null, and then I read the word I combine the two together, my machine does processing, and then spits out a new vector. Then I have this new context representation, and then I see the word have, and again, I'm gonna get a new vector, and I will continue kind of combining the context that I'm constructing incrementally and new words until I finally build my sentence as a vector. And the advantage of this construction is that, for instance, I can take the sentence as a vector and train it as part of some neural model for some tasks that I want to predict. So we are constructing a new vector representation which accumulates context and constructs that context in a way which is beneficial for my application. And to make it, to show you how powerful is its construct, I just want to spend one slide talking about machine translation that you are using in Google. So what the today's machine translation models do, the one that again you use on a daily basis if you are translating from some language, is they go and they read the sentence, let's say in English, okay, create this vector representation, and then they have something which is called decoder, which takes this vector and unrolls it and generates a sentence in Spanish. And the models are trained given parallel sentences of English and Spanish, and it plans how to do this encoding into a vector and decoding of the vector into a Spanish vector directly from this data. This is actually pretty remarkable. And this kind of models are called sequence-to-sequence model because they capture kind of linear context. Okay? So what I wanted to say, I want to go back to the questions that we originally have, which is how can we encode molecules in the smart way? And you can say we already got something when we took Morgan fingerprint, ran neural network and took the first layer. The problem with that is that you are already starting with Morgan fingerprint, which already loses information. Maybe there is something in your molecule that is really important for the task, but it's not captured well in Morgan fingerprint, and really you cannot, you will not see. So what we will do, and I will illustrate you just one example that we developed here at MIT of how you can be constructing a neural representation of the molecule. Okay, so and again, I told you what you've seen so far is Morgan fingerprint, where we start with explicit structure representation. We don't want to do it. The idea here is the following. What we will do, we will start with a representation of atom, which would use some fixed properties of every atom, okay? So this will be the beginning of the representation. But then we will find a way to collect information from your neighbors, to aggregate them together in such a way that every atom not only remembers its own properties, but kind of knows in which neighborhood it is located. And we will iteratively construct this complex representation of the molecule. Okay? So the starting point of this representation is you take every atom and you record some features of this atom. So for instance, you can obviously have atom identity, degree, whether it is in ring, and so on. So you will have a small feature vector that represents every atom. The next state, what you're going to do, you're going to take this atom and you're going to take the representation of the neighboring atoms, okay, of its immediate neighbors, sum them up. And now this will be our immediate neighborhood. And then you will concatenate them together and learn how to aggregate them using the neural net, meaning that you have some prediction task, let's say toxicity. So the way how these pieces are going to be combined together will be driven by the final task that you are trying to predict. So at every single point, again, you're taking your own representation, you're taking the representation of your neighbors, and the way they're aggregated will be driven on the prediction on the final task. And we will go through multiple iterations, incrementally taking contribution from further remote neighbors, correct? Because your neighbors after every single iteration will change the same way as you are changing. So in this case, what we're actually getting is a representation which is task specific and which captures the context, which is driven by your neighborhood. And the final thing that I want to say, in order to get the representation of the molecule, for instance, we can take the representation of all these computed atoms and sum them up. And we've demonstrated in the experiment that it actually performs pretty well in multiple property prediction tasks. So this detail, it's like quite elaborate papers that was published in machine learning conference, like the details here may be less important. What is important is to understand that now we have mechanisms in a smart way to construct this representation, and we are not limited to the representation that many of you are familiar with. We can think of a novel way when we understand what properties we care about to create models which can capitalize on them. So now we got to the end of the tutorial, and I hope you got some ideas how you can apply machine learning in your daily work. Thank you.
So I think you saw the video how Professor Regina from MIT, like she explained in a very simple way about how these neural networks work, okay? And she gave several examples, and then she tried to connect with the, feature extraction from molecules and how to actually use deep learning neural network for predicting properties of molecules. Okay, now coming to the different tools that this group has developed. I told for synthetic planning, you have a tool called ASCOS. Okay, suppose you want to synthesize a drug. Okay, so people nowadays they design the drug using computational resources, and once they design the drug, they want to do retrosynthesis. So now they know this is the drug to synthesize, and now they have to chalk out a way by which they have to do a retrosynthesis from some set of reagents. So they have to optimize, like what are the best ingredients to use, what are the best starting material to use for synthesizing such a product, and how many steps are involved, and then what are the possible side reactions that can happen, and how to avoid them. So all these things are part of synthetic planning, and this group has got a tool called ASCOS, okay, to help in that. So as the website says, I think you're able to see, right? They have got a number of computational tools in assisting in synthetic planning and other aspects of organic chemistry. So here you can go and get started here. Okay, as I told, there's a retrosynthesis. Retrosynthesis means like you have designed a drug and then you want to chalk out a strategy to synthesize that particular drug. So you want to go backwards, like this is the final product, and then like I have to work out a way to find out the starting materials. Okay, from which how do you start? For example, you would take aspirin. Aspirin, how it's made. Like what is the cheapest material, starting material can take to synthesize aspirin? Aspirin is acetyl salicylic acid. So if you want to synthesize aspirin, there are various. For example, I can take different reagents and combine and form aspirin. But which one will be economically the best? and which one will be free from side reactions, and because purifying the product is also a lot of costs involved. So I don't want side reactions and byproducts in the reaction. So to avoid all those things, I need to chalk out a strategy which works well. So aspirin can be synthesized how? So the thing is, I can take phenol, I can take salicylic acid, I can take. So there are different possibilities are there. But which is the easiest raw material to use? That is, we have to take phenol, and then we have to use carbon dioxide, use coal-based reaction, and then salicylic acid will be formed. That is actually treated with acetic anhydride, and then finally acetyl salicylic acid is formed. So this is the one synthetic route. So we need aspirin, and then we go backwards and find out what is the raw material to use. The raw material will be simply carbon dioxide and phenol. Okay, carbon dioxide and phenol, when they combine in coal-based reaction, they give you salicylic acid, which upon acetic anhydride treatment gives you acetyl salicylic acid. Like this, we can actually come up with a synthetic plan for a target molecule. The target molecule, for example, acetyl salicylic is a very simple molecule, but when you have a complex molecule, like for example, you see the structure of lopinavir, such a complex structure, we will not really have any idea about how to start with, which is the best synthetic plant to use. So because of the complexity of the structure involved, you see the structure of lopinavir. So it's quite a big drug. Understood? Nah? Like even though it's within the 500 daltons, when you compare it to the aspirin or any other paracetamol, this drug is quite it has several bonds, some chiral bonds, and then it has several ring structures. So where to start with, how to start with, we do not know. So that is why a synthetic planning may be required. Okay. So the thing is, so this is for normal molecules, so which you have designed in your computer and you want to synthesize it. So you can use these modules. Okay. So they have got a tree builder also, like where you can have several possible parallel reactions happening, and you can choose which one is the best. Okay. So thing is, like these are all available. The entire thing is available in actually GitHub. Okay, so you can go and to GitHub. So you can see the results. I think to log into the site, we need an MIT credentials. Okay. This is for their own group. Yeah. So here, this is the one-step retrosynthesis thing. They've got a simple, examples. Okay. So here you can just paste the smiles format. You can even draw the structure here. Okay. So, for example, I want to synthesize this lopinavir, or let us start a simple structure initially. So what I do, I just put this ring. This is aromatic ring, and then what I do, I add a phenol group. Okay, so this is phenol. So phenol group is there, and then I can attach a COH group. So COH, yeah. Put C. Where's C? It's here. Okay, so what is this one? Any idea from participants? Hello? This is salicylic acid. Okay, So you have OH group and the carboxyl group COOH. So if you have COOH and OH group close by, okay, ortho to one another, that is called salicylic acid. Okay. Now there are two possibilities. Okay. So yeah, salicylic acid. So the thing is, now what you can do is, if you want to synthesize aspirin, okay, a simple, I'm starting a simple molecule so you can understand. Okay, so you want aspirin to be made. Aspirin is acetyl salicylic acid. That means this OH group has to be made into a acetyl group. Okay, so it will be like this. So I have to put C here, and then I have to put a double bond here, and then a single bond here. And then a CH3. Okay, so this is what is this one? This is aspirin. So the phenolic group I have made into a acetyl group. So it's a OCOCH3. Okay, so you can just draw, like okay, this is actually not a bond here. Since we draw close to another, it became like a bond. We can put a bond here. Okay, so what I'm going to do is, because the molecules, we have to draw a little bit this side. Put here, and then put double bond here. Then this O, and then put this CH3. So this is acetyl group, CH3COO. Okay, so this acetyl, the phenolic group has been acylated. Now the OH group you have to take care. Na? So what I do, I put, no. One second. It's like easy to draw, but sometimes they have to be when they're close by now, some problem comes. So I put first to draw the carboxyl group, and then put one O, then I put oxygen. Okay, so I draw the acid part. Now the phenol part I have to worry about. You put like this. CO. So now double bond. Close by doesn't work. Oh, okay. So double bond. Okay, so here I have to type double bond. single bond. Okay, so I managed to draw. So this is actually the structure of salicylic acid, acetyl salicylic acid. So I acylated the OH group, phenolic OH group, so it becomes acetyl salicylic acid. Okay, so acetyl salicylic acid is nothing but aspirin. So if you want to synthesize acetyl salicylic acid, what are the best raw material? Okay, so this is a target compound. I'm just done the drawing. Now it's automatically converts in Smiles format. The best thing for you, drawing, just I did to demonstrate drawing, I just drew it. Sometimes drawing very close by atoms, now they it will be slightly difficult. So if you have Smiles format, now you can just copy and paste it. Okay, you can go to drug bank and you can get the Smiles format for this same thing, and you can get it pasted here. So that is the most easiest option. So the type aspirant. Yeah, you can see this acylated COC3. Yeah, same thing. Okay, so what you do is you go here, take the Smiles format, and the Smiles format you can see the same thing is there. This is the Smiles format. That is what is also here. Can you see? See the same thing. Okay. Okay, good. So now we have drawn this one, and yeah, so it's the same thing there. Okay. Now, if you want, you know, you can just paste the same thing. What is there? Copy here, and then paste it here. Okay. Now we'll use default settings. Template prioritization. Apply all relevant templates. Number of templates 100. Maximum cumulative probability is 0.995. Precursor scoring. Yes, we can go for relevance plus heuristic scoring. Minimum plausibility is 0.75. Okay, search. So this is the target molecule. Can you see here? You just redrawn the same thing. And then the basic thing is the OH group which is getting acylated, and the COH group, they should be ortho to one another. And that is what. So finally it says that the different ranks it's giving. Okay, so different possibilities it's giving. Like how to synthesize that. So it comes up with the new approaches also. Like, can you see this one? This is using salicylic acid. Salicylic acid plus, what is this one? Can you identify this molecule, somebody? Somebody who has some basic chemistry knowledge. What is this one? Huh? No, what is it? It's not simply a ketone. No, it's not a ketone. It is nothing but acetic anhydride. Okay. See, if you take two molecules of acetic acid and you remove water, they will form anhydride. So this is CH3COO, again COO CH3. Okay, so this is acetic anhydride. Two molecules of acetic acid, when they are connected by dehydration, when they are actually, when you dehydrate and form a bond between them, that is called acetic anhydride. So acetic anhydride is one of the important raw material for many acylation reactions. Generally, acylation reactions with acetic acid is very poor. So you have to use a powerful reagent like acetyl chloride or acetic anhydride. Okay, little bit of chemistry knowledge is also needed. Generally, people use acetic acid for. If you think acetic acid means, it's wrong. Acetic acid is not a powerful. For example, that the molecule is selecting for acylation. So generally, it should be a acetyl chloride or acetic anhydride. Okay. So the thing is, whenever you have like some electronegative atom attached, okay, then the CH3CO group, leaving group, will become, you know, like it's easy for it to leave and then go and attack the salicylic acid. Okay, so salicylic acid is acylated by this way. Okay, so this is the easiest option. Yes, of course, the prediction is correct. So we have to take, acetic anhydride and then acylate the salicylic acid. Now, so this is one step. Now the thing is, now salicylic acid, how do I get? How do I get salicylic acid? So again, I can do. So now this acetic anhydride is easily available as a reagent. Salicylic is also available, but I can think of a simpler raw material than salicylic acid. So I can initially synthesize salicylic acid, and then they can actually acylate it. Okay. So what I do, I go here. It gives different steps actually. All possible kind of reactions it gives. Now what I do, I just type salicylic acid here. Okay, this is salicylic acid. So this is one of the stops, one of the intermediates. Okay, so I can synthesize salicylic acid itself actually. So what I'm going to do, I'm going to download a Smiles format for this. It's very simple. So copy this one. So so having understood that aspirin can be synthesized from the top one, this one, rank one, the acetic anhydride plus salicylic acid. Now acetic anhydride is easily available as a reagent, everywhere it's available. So the thing is like easily can be prepared. Now salicylic acid, instead of buying salicylic acid, I can go for even simpler raw materials. So what I do, I just type. So one by step, because this particular software now you can give only one step retrosynthesis. If you want complete retrosynthesis, means it's a commercial and you have to pay for that. It's not freely available. Okay, so that's why I'm using it one by one. So how to synthesize salicylic acid? So this is salicylic acid. So it gives different possibilities. Okay. So you can actually take it from methyl salicylate. Okay, can you see this one? Methyl salicylate. Methyl salicylate is naturally available. If you see what is the difference between methyl salicylate and aspirin? In methyl salicylate, okay, methyl salicylate is actually what happens. The acid group of salicylic acid is actually forming ester with the methanol. So you have methyl salicylate. Okay, see the OH group is free. So the thing is, since the phenolic OH group is free, in methyl salicylate. Whereas in aspirin, it's the other way. The OH group is getting acylated. So and the carboxyl group is free. If we see, the carboxyl group is actually a very mild acid, but the phenolic OH group is highly corrosive. And that is why methyl salicylate is a highly cannot be taken inside. It's highly corrosive. Methyl salicylate now what we apply in amritanjan or this the other things, amritanjan, Vicks, Aprabanol, na. So it's actually this methyl salicylate, otherwise called as oil of wintergreen. It is there. It's a naturally extracted compound actually. From plants you can extract methyl salicylate. Okay, it's called oil of wintergreen. So the thing is, this methyl salicylate having a phenolic OH group, okay, that is actually highly corrosive. So that is why methyl salicylate cannot be taken internally. If you take internally, there is a severe poisoning. So to avoid that, people use it in topical applications. Only externally, the skin they apply. So even in the skin, it causes irritation actually. So since oil of wintergreen can be easily got from plants, okay, so it suggests, okay, you can get the oil of wintergreen from there, and then you can remove the methanol. So demethylation. So what you do, you remove methanol. The ester is broken, methanol is removed, you get salicylic acid. Okay, so like that it gives the option. So if we come here, it throws another interesting thing. Can you see here? Can you see this one? What is this one? Carbon dioxide. COO, carbon dioxide plus phenol. This is what I was telling you. Okay. So thing is, suppose your oil of wintergreen, okay, you can, but extracting it in large scale is not possible. Okay, so we have to sometimes use the other economical things also into consideration. And like who will buy aspirin and break it to salicylic acid? Because after all, we wanted salicylic acid, na? So thing is, this is the best option. So anyway, the score is quite high. You can see the filter score. So here, can you see carbon dioxide with phenol under high pressure? If you take, because carbon dioxide is a gas and phenol being a liquid, you want to mix them, you have to put in high pressure, and then the Kolbe's reaction starts. Okay, and that will give you salicylic acid. Now salicylic acid can be made into aspirin by adding acetic anhydride. Like this, I can use this one-step retrosynthesis tool to work backwards to find out the simplest raw materials. Okay, so if we go and see Kolbe's reaction, it's a +2 chemistry book actually. I'm giving the simplest examples actually. Can you see here? So phenol is made into it's actually like a alkali is added, and then you add neutralize alkali and add the carbon dioxide. So basically, phenol plus carbon dioxide only is reacting. The COO group goes and attaches here, the COH. Okay, so this is how salicylic acid is prepared. So the simplest raw material will be phenol is easily available in large amounts. You take phenol, you under pressure you carry out the reaction with carbon dioxide, you get salicylic acid. Salicylic acid you add acetic anhydride, it becomes aspirin. Okay, it's a very easy way to synthesize. Now the other way you want to do, for example, you want to synthesize methyl salicylate. It is available naturally, but you want to synthesize it. What you have to do? You have to do the other way. Okay, so there what you have to do, you have to add methanol. Okay, so you take up to here it's same, phenol plus carbon dioxide gives salicylic acid, but salicylic acid with methanol, okay, will give you methyl salicylate. So that's the ester between the OH group of the COH group of your salicylic acid and the OH group of methanol. Okay, so that is how like you can use this tool, one-step retrosynthesis tool. So maybe like we can try for the complex molecules also. Having done for the simple molecule, we can try for now we understood how it works. Now what you can do, we can go for a slightly complex molecule. For example, remdesivir. Remdesivir or lopinavir. So you can see here, it's quite a big molecule. You see here, it's very complex. Remdesivir. Its synthesis is not so easy. That is why only few pharma companies are manufacturing this one. Okay, and the cost also is high. Now, what we do, copy this one, and then we can go to this one-step synthesis and paste here. Okay, now default settings. Now, what you can do, we can search. So, can you see? It's quite a complex molecule. Remdesivir. Okay, so this is remdesivir, and then like viability. For example, certain ingredients you cannot buy, so you have to manufacture it actually. So it shows that synthetic options are very few. So you see, these things themselves have to be prepared. Precursor itself has to be prepared. We cannot buy it. No vendor is available to sell it. So this has to be prepared, and then you have to prepare this also. Then you make them react. You will give this one. So in order to prepare this and this, separate synthetic studies are required. So okay, so what you will do? How to synthesize this precursor and this precursor. So what we'll do? We'll just copy this one. Copy and paste it here. So now we are going to synthesize the precursor. What is the raw material? Because this is a very complex, quite a complex molecule. Like it's not so easy. So I'm just now involved in making the precursor. So the precursor itself, you can see now, some are viable. For example, these two are viable, but this one is it cannot be bought. That means you have to synthesize this one in situ. You have to come up with a synthetic strategy yourself. So we have to synthesize this one. These two can be bought from commercial vendors. It's already available. Okay. So what I do, I have to find a way to synthesize this one. So I can paste this one. Okay. So default settings, and then I search. Once you become an expert user, now you can change the other ones. Like we are using default versions right now, but now you see, now these two are available. Okay, from vendors. So they can be bought. They can be bought. So it's now, this is how we have to proceed for retrosynthesis. So you have to buy these two, and then you have to, because these are available from vendors. Suppose it's costly, and then you can make yourself again. So suppose this molecule buying option is very costly. Then what you can do, you can try to synthesize that also on simpler raw materials. So like this, one step, we go backwards and find out the best raw materials to use. So this, yeah, so you have to just like an ester form is available. So you have to de-esterify and use it. Okay. So I think this is precursor simple only. So like this, step by step you have to go. Okay. One step retrosynthesis. Yeah. Come to remdesivir. So again, coming back to remdesivir. So we saw how one of the precursors of remdesivir or manufacture remdesivir can be this molecule, this precursor, how it can be actually, like going back several steps, how we can use simpler available raw materials which are available as chemicals from vendors. So we can chop out a synthetic strategy. So even if you are not a very good chemist, you can use this one because it's such a complex molecule. We really do not know where to start with. And even experienced chemists, they may have some difficulty. So here it simplifies the problem. So the thing is like, now this one, you'll see this precursor. So one of the precursors of remdesivir, this one which we saw, like how to go backwards and find out.
Somebody is asking what is score represents.
Which one, sir?
Somebody is asking about the score. What does the score represent? Here the score is in negative.
Yeah, this is actually like you have to see this filter score only. Like the thing is like score the sense like synthetic possibility, manufacturing possibility. For example, how easy it is. It is not very simple. If we are able to buy now, it's very simple. But the thing is like finally you can see the score. This is the score to see. There are a lot of things there, template driven. So you don't consider this one. Finally, see this one, fast filter score. That means, like among the different alternate possible reactions, which you can, for example, I can get these two precursors, and I can go to this one. So which one gives me the best one, like the best possibility? So I have to see this one, suppose these two precursors have been used, there's a better chance of getting this one, remdesivir, without having other problems like side reactions and for example, now to carry out a chemical reaction, there are many things you need to like some are very extremely difficult. Okay, so this is actually the score shows how the score actually, since it's can you see now it's a negative, and if you see the ones which are actually like more complex, okay, the score is even becoming a larger value, but it's negative larger value. So what it means? That means synthetic difficulty is very, very high. So the score is actually the the ease with which the synthesis can be made. Okay, so actually remdesivir is not so easy. That's why even the precursors, na, can you see, this is not a very easy. So that is why the score is negative, and then it has some kind of quite large negative value. That means it's not a simple thing to manufacture these precursors. Okay, that is why we have to go several steps. It's not a simple one-step retrosynthesis. Okay, so but we are using this tool because one step we go backwards and find out the thing. Okay, so what I can say is the score represents how easy it is to actually carry out this reaction. Okay, so if we compare this one, this will be more difficult to carry out this reaction using these precursors to get the structure of remdesivir finally. Okay, if we go to this third option, it will be even more difficult. You can see the negative value increases -9072. So actually, it's not an easy reaction. It's not a simple cakewalk to manufacture remdesivir in lab. It will be a multi-step reaction, and it will be quite difficult to manufacture. That is what is actually showing here. Okay, so it's actually the ease at which the drug can be actually synthesized using those precursors. Okay, so that means like compared to other options, this is the easiest option. So what you can do, this itself can be actually prepared. Since you cannot buy this one, so you have to prepare it actually. So like this, now we saw how to prepare this one. We can now go for this one. So I just copy this one. Sorry, I have to copy this one and go here. So this is one of the precursors. Okay, so the precursors generally I think it's very difficult to buy. You cannot buy this one. No buying option. The only thing is you have to use this one. I think this is even more. Yeah. So here you can see all these OH groups. Na, they are actually like oscillated. Can you see? This group is oscillated. This group is oscillated. This group is oscillated. So this form is a precursor, whereupon you can de-escalate and then use it. Okay. But anyway, like manufacturing this one will also be like difficult. That is why, like, I think only this company called Hetrona, there is one company called Hetro, that is only manufacturing remdesivir in India. They only got the license to manufacture remdesivir in India. Otherwise, all are imported actually. I'm just giving a very complex molecule, so, just to know whether this particular server is able to, Yeah, now you can see. Like no, but again it comes back to the same thing. This one. This cannot completely replace the synthetic chemist, but it can help him to actually, select a particular strategy over the other. Okay, because there are many things to be considered. So the thing is like it cannot replace the synthetic chemist completely, but it can actually like help him, rather assist him in actually in retrosynthesis. Okay. So now, what we can do, like you understood now, like we took a complex molecule, not everything can be done using this one. But it's a one-step reaction. One step, you can go backwards and you can go. Now,
So what is asking about fast filter score, sir? Fast filter score.
Yeah, fast filter score is finally it's a translated score. See, actually, like the thing is, like if you want to read the exact meaning of how this is arrived, it's using some equations, because this particular author would have given some equation to calculate the score. Okay, so you have to go and read that one. But anyway, like out of my understanding, what I'm saying is the score represents how difficult it is to synthesize. Okay, if it's a positive score, means what? I think it's quite easy. For example, when we go for this, for example, we take this aspirin or something. Aspirin. Okay, so you got aspirin, is a very simple molecule, and which is well understood. Like any company in India can manufacture aspirin. So it's a very simple strategy. So what I do, I just type aspirin here. I just remove remdesivir and put aspirin there. Okay. Now what I do, search. Can you see here the score? Okay, it has become a very, very small one. Okay, that means it's possible. It is easily possible. So the ease of synthesis is there. Okay, because last time what you saw minus and then some huge, some several thousands you saw, minus 5,000 something you saw. That means it's, because negative value, that means it's that is not very easy to carry out the reaction. Okay, it's a bit difficult.
Is there any range for filter score, sir? Somebody is asking.
Yeah, the filter score is a relative score. The fast filter score is a relative score.
So,
like compared to the other ones, like for example, compared to the other options, this is the best one. Understood, na? So it's a relative score, the fast filter score.
okay, sir. Continue.
So just 1 second, sir. I have some courier at the doorstep.
No problem, sir.
Hello, sir. Hello. சொல்லுங்க வந்தீங்களா? 1 second, sir. 1 second. Hello. Sorry for the intervention.
Sir, no problem. You can continue, sir.
So you can always go to tutorial. See, there's a fact and tutorial. Like this kind of questions, you can go and like what is this? Like for example, we just use this application retrosynthesis. You want a multi-step retrosynthesis, like the one for very difficult like remdesivir and all those things. You can use a tree builder. Okay, so there are different recommendations given, and yeah. So you can see the score, the scoring function. So the scoring function, what it says. So select precursor scoring function, scoring option. So here actually, as far as I know, it's basically like how difficult, how easy it is to manufacture. So if you get a negative score, means it's going to be difficult to manufacture, difficult to synthesize that one. So that is what actually I can understand from this one. You can see for these kind of molecules, it's you can see that here the score is minus 1.98, here minus 786. That means this is actually not the best option because you cannot buy it. It's not whichever is not available for you to actually carry out. So you cannot buy that one. That's why it's a negative score. Since it's a one-step reaction, so it says that, okay, the raw materials are not available. So what you will do? Like that it's saying. That's why the score is negative, high negative value. Okay. So the feasibility, basically, we have the raw material available, we can carry out. If the raw material is not available, since it's a one-step retrosynthesis server, it says that, okay, this raw material is not available, you cannot buy it, but these are the raw materials. That means like it will be not possible to carry out the reaction now because raw material is not available. So you have to go backwards again. That is why the score is actually poor. So now coming to the tree builder. So we'll use the next one, tree builder. Modules, yeah, tree builder, path planning. So here I have to actually, the thing is, we have to register actually. And I think last time when I registered, I think somebody's needed. Okay. So here username. Okay, now actually it's simple registration. Okay, tree builder. So here tree builder means you have to, there's one click here to build the whole depth first search pattern. Target compound, okay. So here target compound we can paste it. For example, this time I will start with simple ones. We'll start with the aspirin. Okay, so this is aspirin, and then we will use default settings. It will take some time. So the maximum depth means here how many number of steps this sequential reaction you want to carry out. Okay, so you are given up to 4 here. Because a simple molecule na 4 is enough. So this one minimal retro template count. What is the minimal template popularity you would like to use during the retrosynthetic expansion. In the sense that how commonly it's available. I highly recommend leaving this at 0 unless you have strong reason to only examine the most common chemistries. So the thing is like how, like for example, certain things now like well-known and well-characterized ones. Okay, so the thing is you can go count here, but if you want to explore all possibilities, leave it 0. Number of templates that you can put a large number. Like you will try to take several, precursors and try. Maximum cumulative probability. The relevance prioritizer assigns a probability to each template based on the current target molecule for the product to be formed. Templates are applied in decreasing relevance. What is the, so that means the ones which are highly relevant is the ones we initially tried. What is the maximum cumulative probability you would like to reach before templates are no longer applied? So that's why 0.993. Now the branching factor. What is the maximum branching ratio to use during expansion? For example, you remember it's like a tree, to go backwards and find the raw material. It will be like a tree, like several small molecules that will be combined to make one semi-precursor that should be combined with the other one to form another precursor. Finally, the end product is one. Okay, so the thing is, you will have like a pyramid. Okay, so the top of the pyramid will be your molecule which you want to synthesize, and then the base of the pyramid you will have several precursors which have to be connected, and then the molecules formed. Okay, so the expansion time. How long do you like to let the expansion run for? So here actually it depends upon how quick you want to get results. So if you want to explore more, then you have to give up to 600 seconds. 600 seconds for very exhaustive search. Okay. Super small, depending on the complexity of the molecule. There's a small molecule like aspirin, 30 seconds more than enough. So here the score is given, and they say, okay, this is your acetyl salicylic acid aspirin, and then the raw material should be like acetic anhydride and salicylic acid. Okay. Now salicylic acid. Now they taken salicylic acid. Acetic anhydride is easily available from market, so it's very cheap also. So you can get it actually. But at least for synthetic chemists, it's easy to get. But private users now you cannot get actually, because acetic anhydride is banned for private persons, because acetic anhydride can be even used to make heroin. We can morphine from plants can be extracted. One can extract morphine from plant, this opioid plant, pawpaw seeds. You can extract the latex, and then you can extract morphine. And then when morphine is treated with acetic anhydride, it will give you heroin, one of the very costliest opioids. So the thing is, that's why it's not it's available. It's quite cheap, but it's actually you need a license to get acetic anhydride. Anyway, it's available for chemical chemists or involved in manufacturing. Okay. So now if we come to this salicylic acid, this is the complex molecule. So again, you can go for a next step. So here it says, okay, you want to synthesize this one, then these are the possibilities. Okay. So this is our first option. The second option. First option means like you can use acetic anhydride and salicylic acid, or you can use acetyl chloride. I already told you now, acetyl chloride. So acetyl chloride and salicylic acid. Okay. So it's actually like it's augmenting. See this groups. Why you can ask me, sir? Why if you see here now, this option? Can you see option 3? Generally we consider option 3 to the best one if you don't know chemistry. Okay. So we think if you want to get acetyl salicylic acid, take acetic acid and treat with the salicylic acid, you will get acetyl salicylic acid. But why it is not given option 1? It is why it's given option 3. It is because this reaction will not go to completion. Okay, so the thing is, the machine learning algorithm is smart enough to know which reaction occurs much better. This always proceeds to completion and is in quicker time also. So it's not only economics of that raw material, it's also the chemistry also involved. Because when you take this acetic anhydride, okay, this group, this is the group which is actually the leaving group, okay, C3CO group, which goes and oscillates the OH group of your salicylic acid. Okay, so the remaining group actually augments the electrophilic character of this acetyl group. Okay, so if we take the option 2, okay, so compared to acetic acid, acetyl chloride is better, because again this chlorine, now electronegative chlorine, it augments the electrophilic character. It improves the electrophilic character. You know what's electrophile? Electron loving. Okay, it's because you are if you take your ring, aromatic ring, here you have to target this one. Okay, so the thing is here the augmenting the what electrophilic character of the acetyl group. So that is why the chlorine or the anhydride is important. Okay, rather than the acetic acid itself. So this means that this particular tool, machine learning tool, not only is just considering economics and feasibility and all things, but also the reaction chemistry is also it's considering, which is the best one, which will lead to completion of the reaction. Generally, when you use acetic acid plus salicylic acid, the yield is very poor. The aspirin yield is very poor. Okay, compared with when you treat the acetic anhydride and acetic acid, we will talk about it. Okay, so it's able to predict that one. Understood? Now it's able to predict. That is the good thing. Okay, so now the many options are there. Now what we can do? So now since it's a multi-step, so it gives hundreds of options. You see. Now we'll go to the next one. Yeah, it gives several possibilities. Yeah, several options it gives. More easiest ones to all type of possibilities it gives. Okay, so now coming here. Coming to option 1, so we'll restrict to option 1 now. Okay, if it's green means it's available from the vendor. If it's red means you have to synthesize it. Now, salicylic acid is available, but you want to synthesize this target. That is also possible. Synthesize this target. So now we are trying to synthesize which one? The salicylic acid itself. Okay, so use default settings and then you come and start. Yes, this ASCOS is a free tool only. Okay. Most of the things I'm explaining out free tools only actually. That students can go and use it. So it shows, can you see? Now it shows the best option. Can you see here option 1? I told now. Carbon dioxide plus phenol. Phenol generally should be in the phenoxide form. Phenol means if we treat with some alkali, like we add sodium hydroxide, it becomes phenoxide. Okay, that means the proton is dissociated. So the phenoxide is actually the one which can actually help in the CO2 group from getting attached to the ortho position. Okay, so the CO2 group actually occupies the ortho position to the phenoxide ion. This is the O minus. Okay, so first what you have to do, you have to take the phenol and make it to phenoxide. It's very simple. Just dissolve the phenol in a alkali solution. That will give phenoxide. Okay, so phenoxide, if you the reason why phenol is very corrosive is because of the phenoxide formation only. Because as soon as for example, now you take phenol can, even in water, not only in alkali, even in water, what will happen is phenol can become phenoxide because the hydrogen is dissociable. Okay, that is why even though phenol is actually it looks like it's not an alcohol, it's a phenol. Phenol and alcohols are different. alcohol means the OH group will be attached to some aliphatic carbons. If the OH group is attached directly to the ring, the aromatic ring, then it's called phenol. Okay. So the thing is, because of the resonance stabilization offered by the delocalized pi electrons in the aromatic ring, this can the O minus can stabilize. Okay. If you take the same example, you take ethanol. Ethanol will be very difficult to form ethoxide. Extreme conditions, very difficult conditions only, ethanol will become ethoxide. Okay. It is very difficult to dissociate the OH group. The hydrogen group, the hydrogen atom proton. It's very difficult to dissociate the proton from the alcoholic OH group. Okay, so but the phenolic OH group is easy to remove the proton, and so it becomes a phenoxide. So phenoxide can be readily formed even in water. It forms actually. Take phenol and dissolve in water, it forms phenoxide. We add alkali, little bit alkali, readily it forms phenoxide. So phenoxide you treat with the CO2. So since CO2 is a gas, you have to use some pressurized equipment. Okay. So the thing is like the synthetic manufacturer, they will always have access to this chemical engineers who can design such equipment. Now any kind of reaction which involves gas, one of the reactants is a gas, then you have to use a pressurized container. So you use a CO2 and then phenoxide, and then you carry out the reaction, you get salicylic acid. The salicylic acid you go and then you can make into a aspirin. Okay, now the synthetic strategy is straightforward. Okay, now let us try the same thing for a slightly bigger molecule. Like maybe we can try with the lopinavir. Lopinavir, you know what's lopinavir? Lopinavir is a protease inhibitor. Okay, so it's lopinavir is a COVID-19 main protease inhibitor. In our lab itself, we docked and saw the score was around minus 10 kilocalorie per mole, the binding score. Okay, but now the similar drugs, not lopinavir exactly, but similar to lopinavir, some inhibitors, they in complex with the main protease, that structure is available in PDB. So I'm copying this one and then going here and then coding the representations. So this is the lopinavir. Okay. So if we take lopinavir, what you have to appreciate is, can you see this bonds here? Can you see this bond? CONH bond. Can you see this one? CONH bond. This CONH bond is like a peptide bond. Okay, CONH. So it's this lopinavir is able to inhibit the protease active site, the COVID-19 main protease active site, because it mimics like a peptide. The structure mimics like a peptide because it has a peptide bond and all. Can you see? But this peptide bond is not easily cleavable. So that is why this cannot be cleaved. So that is why it's actually it becomes an inhibitor. Okay. So peptides get cleaved and the protease achieves the activity. But this is a peptide inhibitor. This is a protease inhibitor. So it has peptide-like bonds. Okay. Now what I do, I go here. All these things you understood now. You go and start here. Okay. So all possible. Okay, so this is the lopinavir, and this is the synthetic route, because you see, it's not viable. This is not viable, so you have to synthesize this target. So that is also like shown here. Can you see? like this part is here and the remaining part is here. This part. Okay. These two have to be combined. And this can be actually split into these two. And this is viable. Can you see now? It shows green color. It shows green color. That means you can synthesize. You can get it. It's even the price it quotes per gram. It's $5. It's okay, portable. And then it's but anyway, like quite expensive only because the raw material itself is per gram $5. Now, if you see here, this is also like there. So these two are viable. Okay. But you see this, it's $35 per gram. It's $22 per gram. So you can, so the thing is like, so these two are there, and this one, this part can be got from this part. So here it's too long now. See, like this branching, it's not shown here. Can you see? There's a branching here. It continues from here. Can you see? So you have to combine. What you have to do is maybe like you can set in a different way. You can see this one. Okay. So what they have done is you have to take this one, this particular product, this particular precursor, and combine with this one. Since it's not possible to, maybe the page was not enough here, space was not enough here, they have just left it like this. So that means it is come here. Can you see it's come here? So you have to combine this one. This is the precursor. This precursor with this precursor to get this one. Now this precursor can be got because it's not viable. It's not available as such in by any vendor. You have to synthesize that. So this can be got from this one, which in fact can be got from these two. Okay. So one is viable, the other one is not viable. So you have to get this one. So you have to get this one, convert this one to this one, and then react with this one. So these two are available from vendors. So you have to buy this one. It's 5 gram for $5 per gram. This is also $5 per gram, $3 per gram. Okay, it's for a drug like this importance. It's okay. So you have to get this one, convert to this one. So what they're basically doing, they are doing an oxidation. Can you see here? The OH group is made into a aldehyde group. This is aldehyde, na? Can you see? How many of you say it's aldehyde? Correct, na? Because the end means it will be aldehyde. It will be the middle means it will be ketone. So it's aldehyde. So they are basically carrying out the oxidation reaction. So you have to get this precursor, which is viable. So you have to buy from this one from the vendor, and then we have to make it into a oxidized state. So you have to make it an aldehyde. This aldehyde will react with this compound, which is again viable. You can buy this product. Okay, so you can get this one from the market. So this oxidized product is reacted with this chemical, and then what you get is this one. Okay, and this one. So this is actually basically a type of conjugation. So can you see here now? Where is the conjugation taking place? It's actually a peptide bond formation. Can you see? This is a aldehyde group. Okay, so what happens is this NH2. Can you see this NH2? This NH2 comes and replaces this. So it's actually a, what do you call this one? It's actually like a, I would say it's like a shifts-based formation. Okay, it's a shifts-based formation. Basically, it's, you know, shifts-based. I heard about shifts-based formation. Shifts-based formation involves a carbonyl compound reacting with a amine, a free amine. For example, if we take lysine, lysine is an amino acid present in protein. Lysine can actually readily form shifts-based with the aldehydes. Okay, so this is actually a shifts reaction. Okay, you heard about shifts reaction, means it's a very popular reaction in chemistry. Shifts reaction. Yeah, shifts reaction generally it will involve a aldehyde, a carbonyl compound like aldehyde, and then it will involve an amine. Okay, so you can see here now. So you need an in shifts reaction, not shifts, just shifts reaction. Shifts base, yeah, shifts based reaction. So here generally what you have is actually you will have a yeah, you will have an aldehyde. Can you see an aldehyde reacting with the primary amine? Okay, so then what you'll get? You'll get this one, a carbonyl amine, which can break down to this double bond thing. Okay, so basically this is a shifts reaction. Okay, shifts based formation. Shifts based formation. Yeah. So here aldehyde. So we have this aldehyde, which is got from oxidation of the precursor molecule, and then it treated with the amine, and then you get a carbonyl amine. Okay, this is what is actually shown here. Can you see this is an aldehyde? This is an amine. NH2 group is there. Okay, primary amine. So aldehyde reacts with primary amine to form a shift base. That is what is here. Can you see? This group is attached with this group, and so NH2 has become NH, and this oxygen is actually removed. Okay, so shifts base is formed. Now this shifts base, what it does? Can you see what is happening? So this is actually modified. Okay, this is modified into this one. Can you see this one? This group. Can you see this one? This actually cyclizes. Okay, this is actually removed. This part is removed. That tertiary butyl group. This is the tertiary butyl group. Now can you see? CH3, okay, so this is the tertiary butyl group is removed. It's like a blocking agent. This tertiary butyl group is removed, and then this cyclizes to form this ring structure here. Can you see? These are got from these two nitrogens only. This one. So this part is same here. This part is here, but this part, this tertiary butyl group, the T-BOC group gets it's a in many organic reactions. The T-BOC is actually a blocking agent. It blocks the functional groups from unwanted reactions. So this is removed now, and then it cyclizes to this one. Okay, so you can see now this is like a what you can see. This is like a have you seen this uracil and all? Have you seen uracil? Huh? How many have you seen uracil? It's quite similar to that one. Can you see? The only thing is not exactly uracil, but it's quite similar to the one. It's actually a dihydropyrimidone. Dihydropyrimidone. Okay, so this is getting formed actually. And then this has to be combined. This has to be combined with this one. Okay, and this one itself is actually manufactured from these two. Okay, these two you can get from the market. React with them. So this is conjugated, and then this is actually made to this one. Again, you can see this in a T-BOC group, and the reaction has to happen with the correct functional groups. There are other functional groups which can react and lead to sideward reactions. That is why they block those functional groups, and that NH2 group reacts with this one. Again, can you see here? Okay, so this is a type of an amide formation basically. Can you see here? NH2 with COH group, it forms a peptide bond. Can you see a peptide bond, amide bond? So so this is basically a amide formation. Okay, and this one actually what happens is they are removing the blocking group. The T-BOC group is removed, so it just goes. The entire thing goes here. The T-BOC and the ester group The ester group also breaks here and goes. So now it has become NH2. This NH, this COO, T-block goes off, okay, and then you get NH2 here. So the functional group is again retained, okay, after the reaction. Now this one is needed now. This one is needed to be reacted with this one. Okay, so here this NH2 group is actually involved. Can you see this one? This one is there. So this one is here. Can you see? Can you see this one? This one is here. This one is here. Now this part, COO, this part. Okay, so this part actually is not there. Okay, so this part is removed. Can you see here? So this part is up to here, it is there, and then what happens is this one comes and attaches. This particular NH2 group is involved in interaction. So this NH2 group forms a amide here. Okay, so this is the leaving group. This group leaves, and then like this entire thing leaves, and then this group NH2 group comes and attaches here. So that is why you get this molecule. Okay, so this is how we can chalk out a synthetic strategy for even complex molecules. Okay, so I think you got some idea about how to carry out this kind of complex synthetic reactions. So basically, you can't replace the synthetic chemist completely, but synthetic chemist, he can use it to advantage. Okay, like how doctors can use machine learning tools to their advantage in diagnosis. Nowadays, cancer detection and all those things, they can subject to one more round of verification using machine learning tools. Even though the doctor may have made a small mistake somehow, it can be actually like machine learning tools can actually one more parallel diagnosis can be made, and then you can confirm it better. Similarly, here also, though a synthetic chemist knows the strategies, he has now more options actually. Okay, so whenever you have very complex molecules involved, and it's very difficult to know which is available, which is not available, what is the price of the reagents. There are different features. Okay, like she was explaining the previous tutorial. There are different features to be used, and the most important feature is the reactivity. Okay, so you should not. We can include more and more features to make the machine learning program very sophisticated, but we should not ignore the most important features. Okay, so that is what is actually here. So it's like they've been trained. This particular algorithm has been trained by supervised learning. Okay, supervised learning, and there is something called unsupervised learning. In unsupervised learning, you just feed the data, okay, to that algorithm. The data learns that the data is directly fed to the algorithm. The algorithm learns from the data directly. There's no need for anybody's supervision. So there techniques like PCA, you heard about principal component analysis. That is actually one of the unsupervised learning method. Okay, so which can extract a certain important, for example, whenever you want to do dimensional detection or feature extraction directly in the data, we can use a PCA. Okay, principal component. So that's actually like a way to use unlabeled data and then use PCA and other things and then get it labeled. Okay, so these are different algorithmic approaches available. So now, if you want to know the code, so now this is a web server and you're easily using it. Understood? Now you want to design something like that. You want to get more sophisticated. You want to learn certain programming tools like this Python programming. That to Python programming suitable for machine learning. Okay, and of course you need for this kind of complex things now where you see several reaction outputs are actually monitored. So for these things you need GPU, as she was telling about GPU. Okay, whenever you have deep learning involved, where multiple hidden layers, multiple calculations have been done, then it's better to use GPU. Okay, so all these things will ensure the success of your AI tool that you develop. Okay, so we'll end the today's session here. Tomorrow we can see how we can practically do a simple coding. Okay, simple. For example, if you take this AlphaFold and all those things, not many other machine learning tools, here is gradient descent minimization. Okay, So the stochastic gradient descent minimization, how it can be implemented using a machine learning code, we will see tomorrow. Okay, and many more things which we left now, like dpubchem and all those things. Like that also we can explore tomorrow. Okay, so today can we wind the session?
Yes,
What
yes, sir. Sure.
Yes, yeah, please.
Yes, sure. Thank you, sir.